Thank you very much, everybody, for being here. Thank you, Alice, for the invitation. It's an absolute pleasure to visit this lovely campus on a sunny day. My name is Elisa Galliano. I'm a neuroscientist, and I work at the University of Cambridge, and I have a young lab that studies plasticity. So I'm going to give you an overview of what I have been doing in the past few years. And I really want to start with the most important slide of all, so I'm sure that I have time for it, which is the acknowledgements. So some of the work that I'm going to show you, which is what it's published, and so you can, if you want, go back and read if you're interested, it's mostly originating from my postdoctoral work, which I've done half in London and half in Boston with Matt Grubb and Venki Murthy. And then I'm really going to give you an idea of what this amazing group of young scientists is doing in Cambridge. So in a nutshell, what I'm interested in and what I've been interested in my entire career is how organisms interact with the environment. And to schematize it to the core, well, they receive sensory inputs, they compute them in the brain, and they provide an appropriate behavioral output depending on the situation. Now, if we make this slightly more complicated by adding one single variable, which is time, we know that with the passing of time, the environment changes, and so will the sensory input that the organism receives. So at this point, a binary choice. What can the organism do? Can either change the computation in the brain to maintain the same behavioral output, and this is adaptation and homeostasis is also what in biology is called the Red Queen hypothesis from Alice in Wonderland. You need to change to stay the same. Or can use this different sensory input to change the computation and provide a different behavioral output, which we believe is what underlies learning and memory. What is this computation? What is this changing that I've been referring to that is underlying adaptation and learning? We refer to it as plasticity. And I study neurons, but I'm very well aware that there's glia as well in the brain. And it's very important and is also changing. But if we focus on neuron, I think that what makes them unique is that they produce electrical currents, and these trains of currents carry information. So we like to define plasticity in a very holistic way as any mechanism by which these neurons change their electrical activity, therefore change the information they produce. Of course, the most studied way in which they can change the amount of current that they put out is by changing the amount of current that's put into the neuron at synapses, and synaptic plasticity is considered a prime candidate underlying learning and memory. But I do believe that there are other ways in which neurons can change the amount of current that they put out by changing, for instance, the machinery that generates action potentials, nothing to do with synapses, just voltage activated sodium channels, for instance, or potassium channels, and or by changing the structure of the neuron at synapses or elsewhere, because by changing the structure of a neuron, you change the electrotonic properties of a neuron, therefore its electrical output. So if we try and put this more complete picture of neuronal plasticity into our original schematic of environment and organism interaction, I call organism in my lab mouse because of its tractability. And I like to argue that the best place in a mouse brain to study activity-dependent plasticity is the olfactory bulb. The olfactory bulb is this part here, very superficial, very frontal between the eyes, these two blobs. And it's very tractable in a mouse. It's very easy to manipulate the sensory input, for instance, by removing the sensory input by plugging nostrils, or by simply putting some odors in the cage of the mouse. In terms of circuit, is also very, very tractable. If we start from the environment where you have your odorants, then you go inside the nostril and inside the nose, you'll find in the olfactory epithelium, which is this piece here in yellow, these specialized receptor neurons, which are called olfactory sensory neurons. They got cilia. On top of the cilia, which they protract into the cavity where the mucus sits. They have the olfactory receptors, which bind the odorants and the chemical transduction is achieved. These neurons send their axons through bones inside the brain to then reach the olfactory bulb, where they form glutamatergic synapses on principal neurons, which are called mitral and tufted cells. So this is glutamate. And mitral cells go directly to cortex. Two synapses, and we are from the environment to cortex. And you probably are aware this doesn't happen in any other sensory system, because every other sensory system, at the very least, has a thalamic relay. So to study the loop completely, I really believe that this is a beautiful model, because in two synapses, you are from whatever you do to the mouse to the perception of the odor, and eventually the computation of the behavior. This glutamatergic synapse here in the olfactory bulb is not just a clean relay, it's modulated in a layer which is called glomerular layer, where various processes of very many different interneurons help sharpen the information transfer. And among them, my favorite are dopaminergic interneurons. And this should come maybe as a surprise. There is dopamine outside of the midbrain. Yes, there is. They're very strange neurons. And I thought they were a fantastic candidate to start studying activity-dependent plasticity, because I was told that they are homogeneous population, very easy to target, because of course all the people studying Parkinson made transgenic mouse lines that could be just used simply for olfactory bulb. We know from the literature that they can modulate olfactory behavior. Eventually, we want to close the loop, so we wanted to pick something that actually has an effect. And they are extremely plastic, to the point that they can regenerate the entire neuron throughout life in a process called adult neurogenesis, which, if you wish, is the most extreme form of structural plasticity. You don't put out a new spine, you put out an entire new neuron. And you know, there are very, very, very few neurons that can do that. Some are the granule cells in the hippocampus, and the other ones are all in the olfactory bulb. So very plastic, very important neurons. But I discovered very early on in my postdoc that I've been sold a lie, because they're not homogeneous at all, which is a problem, because when you're trying to modify them, you need to have a stable baseline. But if the population is not homogeneous, you don't know what you're looking at. And so I went down a rabbit hole that resulted in another paper. And to cut a long story short, dopaminergic neurons in the olfactory bulb exist in two subtypes. And they can be cleanly defined depending on their development. The majority of them are indeed capable of adult neurogenesis. They're born in the embryo, but also throughout the life of the animal. A minority of them are born only during embryonic life. Incredibly cleanly, there is a second characteristic that really classify them in a binary way. The ones that regenerate do not have an axon. And this is a big thing for a neuron, not to have an axon. The ones that are born embryonically, they are normal neurons. They do have an axon, as well as dendrites. Morphologically, they're very different. The anaxonic regenerating one, they're tiny, and they're very narrowly branched. The axonic one, born in the embryo, they're large. Scale bar here is 20, here's 10. So very, very big. And the dendritic tree branches further in this glomerular layer. The fact that one type has and doesn't have an axon functionally map onto a different excitability, because not having an axon also means not having an axonal segment, which I'm going to talk about in a moment, nor a lot of highly gathered sodium channels. So these neurons can generate action potentials, but not very many. They can't keep up. The fact that the dendritic tree is differently branched has a big impact when we look at their tuning to odors in vivo. So those are calcium imaging data, and the dots here are different odors. And you see that these guys over here respond to all three odors in different ways, and this one only to the third one. And this is because in the olfactory bulb, odors are encoded in discrete areas. And so if you send dendrites to many, many areas, you'll be participating in more the computations of more orders. So again, very different neurons. And so my whole idea to study integrated neuronal plasticity really had to merge with this study of dopaminergic heterogeneity. And we're currently finalizing a study in the lab led by this marvelous master student, Maggie, with my colleague Sue Jones, in which Maggie was fetching the two dopaminergic subtypes in the bulb, but also in the same experimental conditions, dopaminergic neurons in the nigra and ventral tegmental areas to try and see, do they have anything in common with the one in the midbrain? And you can appreciate that. These guys regenerate throughout life, and these ones tend to die. So it would actually be interesting to know if they have any functional similarities, potentially for, cell replacement therapies or things like that. So these two branches of research is what really is being merging in my lab to try to understand if these dopaminergic neurons in the olfactory bulb that have different development, different morphology, and different physiology also have different plasticity. And because I like to look at the normally overlooked things, we didn't start by looking at synaptic plasticity, but we start by looking at the other two types of plasticity, intrinsic plasticity and structural plasticity. And I have just hinted about this for a minute ago. The perfect place, in my opinion, to look at the work of intrinsic and structural plasticity is the axon initial segment, which, as the name says, is in the proximal segment of the axon. This is not the hillock, is not an anatomical demarcation, it's a molecular demarcation. The axon initial segment is where a huge complex of protein, chiefly anchorin G, keeps in place lots of stuff that links with extracellular matrix, for instance, but chiefly voltage-activated sodium channels. The axon initial segment is where the action potentials are generated because you have a high density of sodium channels in a very compact electrotonic environment, so you generate action potentials there. Very interestingly, one of my postdoc advisors showed in 2010 that this entire raft moves and changes length. So you can understand that if there are fewer or more sodium channels which are closer or more distal from the soma in a structural way, you are going to immediately modulate the ability of this neuron to produce action potential. So does AIS plus, this work, by the way, was done in hippocampal cultures, pyramidal cells. So the first thing that we studied when I joined the lab was, again, in an in vitro assay, to ask, do the dopaminergic neurons in the bulb that do have an axon and do have an axon initial segment show any plasticity at all of their axon initial segment? And so this is how we look at AIS plasticity. Instead of trying to do immunohistochemistry for sodium channels, it's very difficult, very fiddly. We do immunohistochemistry against anchorin G, which is the protein that keeps it in place, and it's been shown very clearly that it's a 1-to-1 ratio, so anchorin G basically equals sodium channel. And dopaminergic neurons are marked by tyrosine hydroxylase. This antibody tyrosine hydroxylase is a rate-limiting enzyme in the synthesis of catecholamines. So you see in baseline, in a nice culture baseline, this dopaminergic neuron has an axon initial segment there, certain length. And what we did then, in a different dish, we depolarized the culture by having high potassium media. So you really mimic extensive activation. Potassium, as you may recall, is normally high inside the neuron and low outside the neuron. And you can already see it from the picture over there, and this is a quantification. In a depolarized state, the axon initial segment of these dopaminergic neurons becomes longer and more proximal, meaning that the dopaminergic neurons probably becomes more excitable. And this doesn't happen in GABA negative, so glutamate positive. This is because it's very difficult to, actually stain for glutamate. Glutamate is an amino acid, everybody has it. But this is a GABA negative neuron, therefore glutamatergic principal cell in the culture. and their axon initial segment goes the other way around. It becomes shorter and more distal. And hopefully, you're thinking, now I know you're not going to talk because we're recording this, but hopefully you're thinking, like, why? Why is this? Because the rules of homeostatic plasticity of keeping a set point, they say, when circuit is overstimulated, you should compensate by becoming less active, like these guys do. Otherwise, you risk excitotoxicity, you risk just going in overdrive. But these dopaminergic neurons, they do the opposite. They make themselves more excitable by making their initial segment longer and more proximal. And so my counter to this is, but where does homeostasis sit? Does it sit in a cell or does it sit in a circuit? Because if it's both and the circuit prevails, these are inhibitory neurons. So when the whole circuit is overstimulated, you may want your inhibitory neurons to work overtime, to maintain the set point, not inside the cell, but in the whole olfactory bulb network. This is the working hypothesis that we have. And of course, this is all very fascinating, but it's in a dish. Does it have any relevance in vivo? Since the beginning of sensory physiology, how have we studied activity-dependent plasticity in the sensory system, dorsal and visual, monocular columns, etc.? We have removed the sensory input. And in the olfactory system is less, Tarantino-like than in vision. We don't have to remove an eye. We don't have to remove the cochlea. Just put a plug into the nose of a mouse to mimic a mild cold. So we make plugs and we put them into the nose of the mouse and we leave them in place for just a day. Much, much less than a cold, yeah, which is normally 5 days at least. The plugs are incredibly fiddly to make. We make them out of, my student hates me for making her do this. We make them out of Tygon tubing and dental floss and suturing thread. The key thing is that the nose of a mouse is very long. So you stuff them in with some Vaseline, not to hurt the animal. But you see the olfactory epithelium is quite deep inside. So the plug does not destroy the epithelium. It just blocks the odors from going in. And after 24 hours, we did some histology to check if indeed, this is the epithelium, the olfactory sensory neurons, the one with the receptors are fine, and they're absolutely fine. They're not dying. This is caspase-3 is a marker of apoptosis. They're not dying. They're not, we check for density and everything. It's really not a pathological thing. This is important. It's just a temporary physiological change in the state of the animal. And so once we know that the manipulation is physiologically viable, and this is work that I've done during my postdoc with a lot of people participating, we really try and start and ask, what are the effects on the olfactory bulb? First of all, is narrowest occlusion, just 1 day of narrowest occlusion, strong enough to do anything at all inside the olfactory bulb? And how did we investigate that? We looked for immediate early genes expression that you may have heard of, like CFOS, to see if actually they are downregulated or upregulated. And then, if this is strong enough to actually elicit anything, does it elicit AIS plasticity in vivo? And we did immunohistochemistry and electrophysiology to then check for intrinsic plasticity as well, both in the principal neurons, the ones that project to cortex, as well as the excitatory interneurons, and in the dopaminergic neurons. Of course, we only looked at the AIS plasticity of the subtype of the dopaminergic neurons which have an axon, but we looked for intrinsic plasticity in both types, because both types can produce action potential. So it's a 3-stage process. Does it work? Does it do anything to excitatory neurons? Does it do anything to inhibitory neurons? Does it work? Dopaminergic neurons here and excitatory neurons here. Dopaminergic neurons, controls and occluded. This is CFOS, CFOS and, in green and tyrosine hydroxylase in blue. You see already from the picture that the CFOS, the activity early gene activity, is downregulated. This is the quantification across 3 mice, 3 controls, and 3 normalized to the controls. Tyrosine hydroxylase, this is actually fairly strange. As an enzyme, is also downregulated by activity because you want to produce less dopamine if you don't need it. And so we can look that while the density of dopaminergic neurons don't change, the brightness, the amount of tyrosine hydroxylases change. So these dopaminergic neurons are switching off, activity early gene and dopamine production. What about the excitatory ones? Both mitral cells, which are the principal ones, and external tufted cells, which are just a type of excitatory interneuron, if you want as a control for the dopaminergic neurons, they needed a different type of excitatory early gene, which is called PS6. But as you can see, again, also those downregulate. This is actually quite important. 24 hours of a nose plug is enough to change the brain. We didn't know this. I mean, normally when people do this stuff, like, I mean, seriously, they were removing an eye and then looking a month later in development during the critical period, right? So this is actually quite interesting to start with. And meaning now that we do have a functional manipulation of the sensory input to change the brain, we can look at structural and intrinsic plasticity. First, in these excitatory neurons. Mitral cells, the principal one, they go to cortex. They look beautiful, these yellow ones. Look how nice their axonal segments are, quite thick, quite proximal. Unfortunately, they do absolutely nothing. And neither does their intrinsic excitability when we patch clamp them. Now, tufted cells, excitatory interneurons, less nice. Both them marked with CCK, which is a specific marker, nor the IS is like a bit, shabby like that. Also do nothing, and neither does their intrinsic excitability. So we are not claiming causation here. This is important because these were not experiments done in the same cell. This is immunohistochemistry in a perfused mouse. This is patch clamp in acute slices. But correlatively, nothing changes in one measure, nothing changes in the other. What happens in dopaminergic neurons? Well, the ones that don't have an axonal segment, unsurprisingly, don't have axonal segment plasticity. The ones that do, very, very consistently, after just 24 hours of deprivation, they shorten their axonal segment by quite a lot. And they decrease their intrinsic excitability. Interestingly, the anaxonic regenerating ones in the same slice don't change their intrinsic excitability at all. So once more, we're not claiming causation, but the fact that the only change that we see in the intrinsic excitability happens in the cells that actually do structural plasticity of their axonal segment is quite suggestive. And now we're going to go a little bit more in what's ongoing in my lab, not as clean and not as finished. But clearly, what's missing is what is happening at synapses. I don't want to ignore them. I did all my PhD studying synaptic plasticity. We're just getting to it. And this is work that is being led by my second year PhD student, Li Wang, who's patching really every cell type in the olfactory bulb and looking at excitatory and inhibitory synapses. And here are some preliminary data from Li. We see once more differential plasticity between the two types of dopaminergic neurons in the same slice, in the same mouse, in the same circuit. After 24 hours of occlusion, the anaxonic one seems to downregulate their excitatory synapses. Inhibitory one upregulates their inhibitory synapses. Think about it again. While this may make sense, we now have the opposite manipulation that we had in cultures. In cultures, we were overdriving the network by putting high potassium in the media. Here, we are downregulating the network. So we want the inhibitory neurons to shut up. not to be overdriving, but to be quiet. Hence, shorter AIS, less intrinsic excitability, more inhibition onto them to silence them even more. And less excitation onto them so that they're not doing anything. We're desperately looking for a modeler to put it all together because I have difficulties grasping it all in my head. But clearly the next experiment that this also is going to happen, we're going to have to do at some point is to look at what happens in vivo when we do the hypotassium experiment, which really means putting odors into the cage of the mouse. Is it flipped? Is it not flipped? So what do we think is happening and how are we proving it? So this is the olfactory network. And these tiny ones are the regenerating anaxonic dopaminergic neurons, and this big guy is the embryonic-born axonic one. I believe that these small neurons that come in and out all the time throughout the life of the animal are producing a very local and very dynamic control of the synapse between olfactory sensory neuron and mitral cells, for instance, to code for this different sensitivity of odors. Is there a lot of strawberry or there's a little strawberry, depending on what you need. These guys, the big ones, which are there all the time, and they do have plasticity, they may provide this mix of stability and plasticity, for instance, for odor discrimination, because we saw that they participate in different odor blocks. So for instance, they could be there to learn to discriminate between strawberry, blackberry, blueberry, gooseberry, and so on and so forth. Sensitivity versus discrimination. Now, how are we proving it? These are our hypotheses. This is what we're doing. This is what we're doing at the moment. Ailey is another PhD student in the lab, just started her second year, and she's tackling the connectivity of these two neurons. We can't infer function without knowing how they're plugged in exactly into the circuit. And she's doing it in two different ways. First, she's doing it anatomically using rabies tracing. I don't know if you've heard this, but rabies virus could be modified so that you infect a cell, and this virus will jump one synapse, only one, presynaptically. So if we infect this cell here with a combination of primer and rabies, which is going to be green and red, and we know that this is a dopaminergic cell because we do a tyrosine hydroxylase staining and it is blue. What you can see is that the rabies will jump one synapse, presynaptic, to label this other guy over here, which we then know will send synapses onto the primed neuron. And Ailey is mapping these across many mice over the olfactory bulb. This is the olfactory tubercle and accessory olfactory nucleus, and they're all over the various olfactory area. And we're really also trying to see if there's anything in other parts of the brain, for instance, in the raphe where serotoninergic inputs are there to modulate, for instance, attention. The second way in which she's going to do this is by doing what is called CRACM, or channelrhodopsin optogenetic assisted circuit mapping. Have you heard of channelrhodopsin optogenetics? Somebody, yes, somebody no. So to start, this is patch clamp. We have this beautiful tripartite system. We can record from mitral cell by placing an electrode in them. We can stimulate the olfactory sensory neuron. We're going to have a synaptic, glutamatergic synaptic input onto mitral cell. And then we can put channelrhodopsin in dopaminergic neurons. Channelrhodopsin is an ion channel that was discovered in algae, which is activated not by voltage and not by a molecule that binds it like glutamate, but is activated by a specific wavelength of light. It doesn't exist in a mouse, but if we engineer a mouse to express it only in the dopaminergic neuron, when we shine light at the correct wavelength, these neurons will be activated. So we have the ability to manipulate neurons and activate them when we want. And so we can, for instance, check. We are recording from mitral cell, we're stimulating olfactory sensory neuron. This is the synapse in black. When we switch on the light, because these guys are inhibitory, we can see that the response becomes smaller. And then we can do all the various pharmacology to really show that this smaller response is due to the release of both dopamine and GABA from these dopaminergic neurons. By playing these games, hopefully we will know which cells in the system talk to the two different types of dopaminergic neurons. The second strand of work that we're doing is to try and go in vivo, because all what I've talked to you about now was done in a slice or ex vivo. And again, I'm going to very briefly go over it. This is in vivo calcium imaging that you may have heard of. So you can open a cranial window on the skull of a mouse, but you anesthetize the mouse, you take away a piece of skull, and you substitute the skull with a cover slip, a piece of glass. So it's closed. Then the mouse gets, medication, wakes up. A couple of days later is absolutely fine. It just happens to have a window in his brain. I know, it's unbelievable how well they recover, really. And the olfactory bulb, as I said at the beginning, is very, very superficial, very dorsal. So if we put the mouse under a 2-photon microscope, and in front of his nose, we puff odors with an olfactometer, this is what we see. These are dopaminergic neurons. An odor comes up, and you see a calcium signal taken from GCAM6, which is a calcium indicator. It really shows the activation of this neuron when the odor is presented. And calcium is a proxy for action potential firing, because you know that when an action potential is fired, calcium is released from the internal store. And because this is chronically implanted, we can look at the mouse in baseline, we can put the plug, we can measure, then we can also look at their recovery. And sometimes I was lucky to find the same neurons in all three configurations. And very nicely, the in vivo data match the ex vivo data. The response becomes to odor, which is this blue bar, becomes smaller after occlusion and recovers after a few days, meaning that it's not habituation, it's not bleaching of the neuron, it's really just plasticity going on. And this is some quantification across 7 mice. A lot of preliminary work I did. So what we want to do with this in vivo work, we want to close the loop. And we want to actually see, okay, this is happening ex vivo, this plasticity is happening ex vivo, it's happening in vivo. What does it mean for the behavior of the mouse? So we're doing olfaction because, and we're looking at dopaminergic neurons because we know that it can modulate the behavior of the mouse. How do we study olfactory behavior in a mouse? We play games which are very similar to Pavlov's games. We do what is called operant conditioning. We train the mouse to associate a couple of odors from a panel of many with a reward. And so the mouse will lick to get the reward only when the correct odors are presented. So you can look at the percentage of correct responses. So 0 in this case is 50% chance. For instance, when the mouse tries to learn to discriminate different concentrations of the same odors to look for sensitivity, say only 1% of ethyl valerate is rewarded and 10% is not. Or, like in this case, discriminate 2 odors and 14 distractors, 2 targets and 14 distractors. And you can see that they get to 90% accuracy in 10 days, and they remember this for 3 months. Very stable behavior. Now, because I don't really like to water or food deprive the animals, not very nice for them, nor for us, for nobody. Jenny Wang is an engineer that joined my lab last year as a research assistant, and she worked very hard to build a system inspired by the one developed by Andreas Schaefer at the Crick, called Autonomous, in which the mice live in this environment in very big groups, and they have food, and they have toys, and they have bedding. And when they want to drink, though, they got to work for it, like we have to work for our food. They have a chip implanted in their neck, like pets, when, for the passport, the RFID chip for the pets, cats and dogs. So when they want to drink, they go upstairs. There's a door that reads the chip, so you know exactly which individual is going into the second compartment and then locks it so that, there's only that mouse. And here, they're doing an olfactory operant conditioning task with the olfactometer that presents the odor. So training is self-initiated. They do it when they feel like it, so we don't have motivation issues. and it's completely not, they're not deprived. They just need, they do it when they want to do it, and then they get the water that they need. And Jenny has now almost finished building this thing, which is quite cool. And we've also added some touch screens and speakers, so we can do auditory and visual operant conditioning as well. So these are my last slides. I'm really wrapping it up. I'm trying to bring it up to the big picture, which is really a two-part message here. I talked today a lot about integrated plasticity, but I also had to talk about dopaminergic heterogeneity. I didn't want to, but I was conned in working on the neuronal heterogeneity. What I didn't tell you, there are two types of dopaminergic neurons that I've told you a lot. What I didn't tell you is that my other PhD student, Sana Garival, in collaboration with the University of Reykjavik, is also looking at the heterogeneity of mitral cells, the principal neurons. So really, I don't know if you followed a little bit the debates in neuroscience, also with sequencing, RNA sequencing, etc. What is a cell type? What is a neuronal type? Is it what Ramon and Cajal defined 100 years ago? Is it based on RNA sequencing? Is it based on physiology? What is a subtype? And when do we stop subtyping? Because, you know, each neuron is individual, same as people, but when is it actually meaningful for us to define them in groups? Basically, what is the unit, computational unit of a circuit? And this is open for debate, really. And the second big message regards, of course, plasticity. I gave you today an example of adaptive plasticity, and where we took away the input by 24-hour plugging the nose. And while mitral cells, I showed you, had no plasticity, dopaminergic neurons employed various different types of plasticity mechanisms and changed. And this really begs the idea, do interneurons, inhibitory interneurons, act as first responders? Are those the ones that as soon as something goes on in the environment, act to, again, at circuit level, maintain the set point? But if that's the case, what happens if the deprivation is longer lived? And this is something else that Lee is working on, just leaving the plug-in for more days. And we're checking, are those mitral cells finally changing? Is this anything having to do with the principal neurons? Stability, what gives adaptive plasticity? But what about change? Change for learning to discriminate different orders, that kind of change. And since I'm here, I'm going to put this shameless plug. We have a studentship available for PhDs, if anybody of you or anybody that you know is interested, to really use this cool new behavioral machine as well as everything else that we have in the lab, to really try to ask what happens in interneurons and in circuits in general when we try and make them learn. In summary, this is what I've been doing for the past 15 years, and what I'm going to have to do for the next 30, I think, until I can retire. I am trying to use a multi-level approach, going from morphology to physiology in vivo and ex vivo and behavior, to answer this question that to me is really the most important that one can ask. How do plastic changes in individual cells of different types influence information processing in the circuit to then change behavior? Thank you very much.